Well, hello, my lovely students. This is a tutorial on what I was looking for or what I am looking for in the tissue segment for the laboratory digital notebook. And so in addition, please make sure that you're putting in your stamped abstract, your stamped protocol, and your pre-lab questions into their associated pages. I'm going to focus on the tissue histology. And so for this first one, going to go over the kidney tissue, which I've given you an image of here. This one down at the bottom is more zoomed out, and this picture up top is roughly where we've boxed in the lower picture. And so in this particular one, I wanted you to, number one, ID the nucleus of a squamous or squamous cell, and those are these really, really flat ones here. And so we pointed out a nucleus. What you're going to notice is that nuclei tend to be stained darker purple or darker pink in a lot of these histology images. So number one was identify the nucleus, so that dark dot right there of a squamous cell, and you'll see that the nucleus is sort of stretched thin and flat. Two, ID the nucleus of a cuboidal cell. So these cells here that look kind of cube-shaped with a very round nucleus in the center, those are cuboidal cells. And again, I'm pointing out that dark dot, that nucleus. And then lastly, ID the cell membrane of a squamous cell. And so instead of pointing at the nucleus, I'm just pointing at where that cell is, and that would be its cell membrane. It's very, very thin, so you're really just pointing at the cell edges, which would be the cell membrane. Moving on to the second one. So this is where we're looking at columnar cells. And so you could see the whole columnar cell, each one of these is a columnar cell. We're looking at the fundus of the stomach for this. And again, I've got a zoomed out 100x magnification image, and that box area is where I've zoomed into up to 400x. Number one asks for us to ID the nucleus of a simple columnar cell. So again, I'm pointing out one of these dark dots, any one of them will do. And then number two is point out the cell membrane. And so here what I'm pointing out is that little thin layer in between the two cells. That's going to be the periphery of the cell or the cell membranes of adjacent cells. That's what I'm pointing to there. Now, this one also asks name a location where you would find simple columnar cells, be as specific as possible. This one, like I said, was taken from the fundus of the stomach. So it's something you'll find a lot of in your digestive tract, particularly in the stomach. Move on to the next one. This one is stratified squamous tissue. So you hopefully notice that we have many, many layers here. Remember that the name squamous from the stratified squamous is coming from the morphology of the apical layer. And so again, those are sort of stretched out, very pancake-like cells. So number one asks us to identify where the layer of epithelium starts approximately and where it ends. And so it is starting actually up in here. These are stratified squamous. Even these here, even if you put your arrow down here, because these cells up top are dead and you're starting to get into the living tissue here, that would be appropriate. That would be fine. And then indicate the nucleus of one of the stratified squamous cells. So any of these cells in here will do. I'm just going to point out, again, the dark center of one of those cells. That's going to be the nucleus. As far as where the layer starts and ends, we can use a bracket. So let me pull a bracket. And we could say, hey, it starts from all the way up here and it goes to all the way down here. So if you want to do that, that's fine. But if you just use that number one arrow to point out where the living cells just start, that would also be appropriate. I'll point out that one there. You can also use your arrows to move these arrows around. The arrows on your keyboard can be moved. And so again, I'm just going to take this bracket, make it darker and thicker in weight. And there we go, where it starts to where it ends for this particular level. Or if you were to tell me that it starts here, I would also accept that, since it's a little unclear that these are actually cells up top. So that is stratified squamous or squamous. Let's move on to stratified cuboidal. So in this one, I asked to circle the region of a whole bunch of the stratified cuboidal. And so I've added a circle here. This circle right here, I've just added in and put the arrow 1 to this. And then I pointed out for number 2, a nucleus of a stratified cuboidal cell. So any of these cells here will do. They are stratified. You're looking down through a whole bunch of these cells, but they are stratified. These are actually sweat glands, the epithelial lining of sweat glands, which is why it looks like a little hole in the center. So yeah. Name a location where you would find this type of epithelial tissue. This is going to be in the dermis of the skin. That is where you will find this type of tissues in the dermis of the skin in association with sweat glands. So you could see that this is the whole layer of skin with the epithelium on top and the dermis down below. We're taking a look at the dermis, zooming in to a part of the dermis, noticing there are some of these hole-like structures, kind of looks like Swiss cheese. Zooming further into that, we can see the details, and then zooming in even further at 1,000 magnification, we can see some of the details of the stratified cuboidal. So again, the dermis layer was where you'll find the sweat glands, and sweat glands are lined with stratified cuboidal cells. Moving on, don't worry about stratified columnar. We're not dealing with that one, but we are dealing with pseudo-stratified columnar. And in particular, this one has cilia on top. So this is ciliated pseudo-stratified columnar cells, called pseudo-stratified because it looks like they're stratified. It looks like there's multiple layers here, but really it's just oddly shaped cells with one has their nucleus up high, another has the next one has its nucleus down low. And because they place their nuclei at different levels, it winds up looking stratified even though it's not. Now, I asked for number 1 for one of these pseudo-stratified cells to be circled, and I've circled one there. In the 1000, I meant 1000, you're not going to be able to see them at 100x. At 1000 magnification, identify the nucleus of one of the cells. So again, any of these darker circles in here will serve that purpose, and so I'll just put mine right at the edge of that nucleus there. And then identify the location of the cilia. The cilia is this hair-like stuff at the apical end of these cells. This is the stuff that is going to help move mucus across and up and out of the trachea. So they are going to be on the apical end of the cell. Moving on, just another image here. So again, of the ciliated pseudostratified tissue here, epithelial tissue, again, we could see the cilia up top. We could see the nuclei at different levels, again, giving it that stratified appearance, even though these are columnar cells. So how is pseudostratified columnar cells different from regular stratified columnar? Well, that's because these nuclei are actually at different levels, but each cell is actually just a really skinny columnar cell. It's not actually stratified. That term pseudo comes from the word fake, or it means fake, and it's fake stratified. So again, it looks stratified because of the placement of all of the nuclei, but it really is not stratified. This cell does go all the way down to the bottom, all the way down to this connective tissue layer at the bottom. There are not additional cells lining underneath. Where would you find this type of epithelium? As I mentioned, in the trachea, you have mucus, which you could see the goblet cells here, these lighter cells. Those are goblet cells secreting mucin to make mucus. And then those cilia on those pseudostratified ciliated columnar cells are sweeping that mucus and all the stuff it traps up and out of your trachea, and you swallow that. So along the trachea and your bronchioles would be a really good example of where you would find this one. Moving on to transitional epithelium. And so in this one, we just ID the nucleus. And again, they're the little darker pink dots in here. Point out any one of those that you would like. Why is this epithelium called transitional? Well, that's because it transitions between more cuboidal, as what we see here is more cuboidal. And as that tissue fills up, as the bladder fills up, they will stretch and they will become more squamous-like. And so where would we find this type of epithelium? Primarily in the bladder and the ureters, but primarily the bladder, because the bladder is going to stretch and these cells will become more squamous-like. And then when the bladder empties, they'll come back into this form, which is more cuboidal-like. So they transition back and forth. So a couple other tissues. So let's deal with nervous tissue, the 3 types of muscle tissue, and then all the connective tissue. So this is a giant multipolar neuron. I'm going to move the arrows aside just really quick, just to explain a couple things. The nucleus is not the dark dot in the center. That's the nucleolus. The nucleus is this lighter area here, this lighter circle. So I'm going to point to just the periphery of that. And then as for the nucleolus, where we make our ribosomes, that is going to be this dark little dot right in there. So what type of tissue does this one come from? Since it's a giant multipolar neuron, this would be in the nervous tissue category. This is a type of neuron. Name a location. You might find this in the brain or the spinal cord would be a good location to name for this. Moving on into muscle cells, it may be a little hard to see it, but I'll try and blow this image up really quick. You could see the striations running across the cell here, cutting it, I guess that'd be longitudinally, but cutting it from one side to the other, not lengthwise, but from one side to the other, we see those striations. I'm going to go ahead and shrink this image back down. So we do see striations in this particular image. So identifying one of the nuclei, we could do that, one of these dark dots, any of them will do, and then point out a striation, just point at any area where you see the striations, and that'll be sufficient. So identify the type of muscle tissue here, provide reasoning. So since we see striations and we also see multiple nuclei per cell, this has to be skeletal muscle. They're also kind of rectangular in shape and they run a really long ways. Those are other clues that this is skeletal muscle tissue, rather than them being branched or having only one nuclei per cell. That would be something that would designate cardiac tissue, which we'll look at in a moment. But those are a couple reasons. Name a location where you'd find this in your bicep muscle. Any one of your skeletal muscles will do. Let's move on to this one. This is smooth muscle. Notice that they kind of all look spindly and there's no striations. Identifying a nucleus in this one's pretty hard. So if you just point to any dark dot in this, that would be sufficient. So this again is smooth muscle, and a location where you would find this would be the lining of the small intestines. So here we actually see the inside of the small intestines. We see some Peyer's patches even. And then around that epithelial tissue on the inside, around on the outside of it is our smooth muscle. And again, that is going to help squeeze whatever is being digested through the small intestine. And lastly is our cardiac muscle tissue. You can tell this because I'm asking for an intercalated disc. I'm going to make this image just a little bit bigger, just to point it out. Any of these dark lines here, those are all intercalated discs. There's one, there's one, there's one, there's one right there, and right there, and right there. Those darker lines are all intercalated discs, really important for the function of cardiac muscle as they allow the electrical signal to pass through smoothly from one cell to the next. So I'm going to do number 2 really quick, identify a nucleus, any of these dark nuclei. Again, we have individual cells here. It's not multinucleated. That's maybe a little hard to tell, but those intercalated discs are a dead giveaway that this is cardiac muscle. So I'm going to point out one of those intercalated discs right there. Again, there's several. There's 1, 2, 3, 4, 5, 6, at least 6 that I can point out right at the get-go. So that is cardiac muscle. You would find this in the walls of the heart, cardiac muscle. All right. So then I had this last one, which is a duplicate of a previous muscle type. And I was asking you to try and tell me what muscle type this is. So again, we see long, rectangular with the striations. Since it has striations, you can rule out smooth muscle. It's not smooth muscle. It has to be cardiac or skeletal. And since we see multiple nuclei, this is and we do not see any intercalated discs. There's no dark bands running across these. This has to be skeletal muscle. So this here is another form, another image of skeletal muscle. All right. Now we're going to move into the big category of connective tissues. So I'm going to start with the base connective tissue that is found in embryos. This is mesenchyme cells. So any one of these little dark dots, that's going to be a nucleus. Why is this tissue important? Well, it is the embryonic origin of all connective tissues. So blood, bone, connective tissue proper between our areolar, our adipose, our reticular, etc., etc. All of those tissues come from mesenchyme. Mesenchyme is laid down in the embryo and eventually specializes into those other types of connective tissue. When in development is this found? So this is found in embryonic development. All right, moving on to the first type of connective tissue. This is a connective tissue proper. There are only 6 types of connective tissue proper. This is areolar tissue. You could tell because it's very loose, and so we're going to designate it as a loose connective tissue proper. Has a bunch of different fibers, and we point out some of those. So number 1 is our elastic fiber. 2 is a fibroblast. Any of these little dots here would be our fibroblast cells. And then these large pink bands in the background, those are our collagen fibers. I've decided to point out this large one right there because I think that one is the most obvious. Where is this typically found? It's found widespread throughout the body, but especially over the surfaces of muscles. That's what makes up our fascia. Okay, moving on to the next connective tissue proper type. So yes, this is connective tissue proper. It is another loose type because there's a lot of space that can be seen, even though that space is actually just a whole bunch of lipid droplets. This is adipose tissue. You could tell by the really tiny nucleus pushed off to the side, big white area that's nice and open. That big white area in the center of the cell, that is the fat droplet. So again, point out any one of these dark little dots, that's going to be our nucleus, and then any of these open spaces is fine for the fat droplet in the middle of the adipose cell. Where is this typically found? Well, you'd find a lot of it in the hypodermis or the subcutaneous layer. It's widespread again throughout the body, but mostly the subcutaneous layer of fat is where this is found. Moving on to our last loose type of connective tissue proper. There are 3 types. There's the areolar, the adipose, and the reticular types of tissue that are loose connective tissue proper. In this, I asked you to identify a reticulocyte. Any of these cells down in here are reticulocytes. This has been taken from the lymph node. The outer layer of the lymph node has this reticular connective tissue. So where is this typically found? Typically, the capsules of lymph nodes and lymph vessels will have this type of tissue. And again, the type of tissue here is reticular tissue, not reticulocyte tissue, reticular tissue. I'm going to skip this one really quick and do the next one. This one here is maybe a bit tricky, but this is the irregular dense connective tissue proper compared to the next one, which is regular dense connective tissue proper, where you see it all lined up and it's kind of wavy. This one has like crisscrossing patterns. It's not everything is running directly in a line. We have some crisscrossing going on there. So this is a type of connective tissue proper, yes, but it is a dense form of connective tissue proper. And in this case, it is irregular since these ones are heading this way and that one's heading this way. And so they crisscross in their pattern, which gives them tensile strength in multiple directions. Now, for that, I've also given you identify the dense irregular tissue, and that is going to be down here. This is actually a picture of epidermis and dermis, so a picture of the integument system, and the dermal layer, the reticular layer of the dermal layer has dense irregular tissue to give it that strength and tensile ability in all directions. So yes, this is a type of connective tissue proper, but it is dense, and where this is found is typically in the dermis of the skin. So same would go, answers would go for this. Dermis of the skin would be an excellent answer for this. All right, so let's continue with the blood connective tissue. I do want to make a note. There is one type of connective tissue I did not give you, and that is elastic connective tissue. I did not give you one of those, so I won't be testing on that particular one. So let's move on to blood. In this, I asked you to identify a erythrocyte, which are the red blood cells, which are the most numerous here. So any one of these large red blood cells will work. I'll point out that large one right there. Thrombocytes, these are platelets. They're these little tiny fragments that we see there. There's one here and there and there and there. So I'm just going to point out that tiny little thrombocyte. And then leukocytes are these odd ones out. This is, I believe, a macrophage of some sort. We'll talk about it when we get into the immunity chapter. So right now isn't the time to really worry about the different types of leukocytes, but these larger, darker ones with the darker interiors, those are leukocytes. This is a type of connective tissue, but it is not a type of connective tissue proper. This is an oddball connective tissue, especially blood, since its plasma is not made by the cells that inhabit it. Whereas this typically found within your blood vessels is where you will find this particular type of tissue. And then our last type of connective tissue here, or not our last type, we're going to go through the 3 types of cartilage, but one of our last oddball types is the bone tissue. And so this is an osteon. You can see the little lamellae, which are the laminations, and I've asked you to identify an osteocyte. Those osteocytes would be inside these dark dots, which are called lacunae. Again, although this is a type of connective tissue, it is not a type of connective tissue proper. Connective tissue proper is the loose or dense ones that we talked about first. So no, this is not a type of connective tissue proper, even though it is a type of connective tissue. Where is this typically found? This is found in your bones. This is compact bone, is where you'll find these osteons. Let's move on to our cartilage. I've asked you in this one, by the way, this one with its really smooth background is hyaline cartilage. It is, although it is a type of connective tissue, it is not connective tissue proper, so no on the connective tissue proper. Again, connective tissue proper is areolar tissue, adipose, reticular tissue, elastic, dense tissue, irregular dense tissue, and regular dense tissue. There's only 6 types of connective tissue proper. So this is not a type of connective tissue proper. It's a little bit different. It is our cartilage tissue, and particularly our hyaline. It has that glassy appearance with a smooth matrix. I've asked you to identify a chondrocyte, which is one of those little dots inside of one of those spaces. I've asked you to ID a lacunae to the best of your ability. Point out one of these empty spaces. Not necessarily the cell inside of it, but the empty space itself. Something like that would be appropriate. And then identify the cellular matrix, which is the background material. Where would you typically find this type of tissue? Your costal cartilage, the cartilage that connects your ribs to your sternum, would be a good example. This particular sample actually was taken from the trachea. Around the trachea, you've got bands of cartilage for support, and it happens to be that it is hyaline cartilage. Next type of cartilage is elastic cartilage. So again, I've asked you to identify a chondrocyte, so one of these dark dots in the middle of the lacunae, and then to identify the lacunae to the best of your ability. And so I just pointed out that empty space right in there. This is elastic cartilage, and again, it is not a type of connective tissue proper, even though it is a type of connective tissue. Where is this typically found? So the elastic cartilage is found in your nose and your earlobes. Yeah, that would be 2 really good examples of where to find those. And then the last type of cartilage to talk about is fibrocartilage. This is, again, a type of connective tissue, but not connective tissue proper. That's reserved again for the 6 that I mentioned previously. And where is this typically found? Fibrocartilage is found in the meniscus of the knee, and the intervertebral discs are really good places where you would find this particular type of connective tissue. That finishes us up for the connective tissue. And lastly, I'll go through the epidermis and dermal layers and some of the details I was looking for here. So I was asking for the epidermis versus the dermis. The epidermis is this top part going all the way down through the bottom of the pink, the stained pink part. And the dermal layer is this lighter bit going all the way down to where it ends. So I bracketed those two and put the arrows next to them. Here I bracketed a particular area of the epidermis, and what I was looking for is for you to label each of the layers. So number one is our stratum corneum. That's the uppermost layer that's being sloughed off. The theoretical location of the stratum lucidum approximated, it'd be about right there, right above the granulosum layer. It's kind of hard to see in this particular specimen, but that is where it would be located. The granular layer, you can see these cells have those granules, have very grainy appearance, and so that is our stratum granulosum. Our spinosum cells of the spine, anywhere in here would be appropriate, and then the very bottom layer, the basal layer of this set of tissue is located right at the edge. You could point it out right here or right here, any of that would be fine, and below that basal layer is going to be the dermal layer or the dermis. So it should go 1, 2, 3, 4, 5, and 6. Then I once again asked for epidermis versus dermis, but this, if you can tell, is our thin skin because we can see all these different follicles, hair follicles. The epidermal layer is really tiny in this one. It's very, very thin because this is thin skin. And the dermal layer is very, very thick, containing all of those different structures from sebaceous glands, like we see there, there, and there. We also see seediferous glands or sweat glands like this one or that one or that one. Haven't asked for that in this particular one. I just wanted you to bracket the dermis and the epidermis. But now we're going to take a look at the different glands. So number one here is identifying a sebaceous gland, and our sweat gland should look like somebody cut through a crazy straw. And you just see the holes. That is our sebaceous gland. And as you can see, here's another sebaceous gland right here in association with the hair follicle. So that one would be just as fine. And again, here's our sweat gland, our coiled sweat gland. So hopefully from that, you can tell what this structure is. Again, we see a whole bunch of holes as if somebody has cut through a crazy straw. So this structure is a sweat gland. And I just want you to put that answer somewhere on this slide. And then what structure is being shown here? Again, the internal components are very white and sort of fatty looking. And again, if you come back to here, this is a sebaceous gland. This is also a set of sebaceous glands. And then lastly, I asked you a few questions. I asked you to compare A and B. So we can notice in A, there are no hair follicles versus in B, there's a whole bunch of hair follicles, and that should be the tip off that A is thick skin, B is thin skin. Thick skin is found only on the palms of the hands and on the soles of the feet, and it doesn't have any of the sebaceous glands or hair. Provide a reasoning as to your answer to A and B. One of the most stark differences is the presence of hair follicles. A does not have any hair follicles, thus it must be thick skin, and B does have them, so B must be thin skin. So that was the reasoning there. Hopefully this tutorial has helped you out. If it did, please consider giving it a thumbs up. Leave a comment if you're having any other difficulties or if you're curious about anything else. And if you have any questions, please come see me in office hours. Thanks, and I'll see you guys later.